This is Patricia Van Arnhem, Executive Editor at Pharmaceutical Technology and Pharmaceutical Technology Europe, and I'm joined today by Dr. Kurt Nielsen, Chief Technology Officer and Senior Vice President of R&D at Catalent Pharma Solutions. Thank you for being with us today. The recent Catalent Pharmaceutical Technology Survey identified the complexity and technical challenges involved in solubility bioavailability enhancement. What factors have contributed to the rise in the number of poorly water-soluble compounds in development, and how has this affected the paradigm for drug development and commercialization?
Well, what's happening today in the discovery process is that the molecules that are coming out of that process, because of the diversity of the targets and the screening process and the way that it's conducted, are leading to poorly soluble drugs coming out of that screening paradigm. So then what happens is you've got molecules that should be active against the targets, but you don't have a good way to put them into solution and then be able to test them, whether that's in a bioassay or an animal model or a human clinical model. So therefore, the enabling step in taking a molecule from the discovery process into the preclinical and clinical process is to be able to solubilize it. And so this becomes a primary focus of the medicinal chemist as well as the formulator to find a way to solubilize that molecule so that it can continue on its pathway for clinical testing.
And what are some of the attended challenges that arise in formulation and drug delivery when working with a poorly soluble compound, such as in optimizing the drug release profile, maintaining stability, achieving the right dosing, things of that nature?
Well, one of the things that's most important is, well, I guess there's two things really that are critically important. You want to have a product that's stable and manufacturable, and of course it's a given that it needs to be bioavailable. So assuming you're able to take care of the solubility challenge with the appropriate technique and the appropriate chemistry, then you're left with those two challenges I mentioned earlier, which are the stability and the manufacturability. So let's take stability. A primary concern is that your molecule is going to be chemically stable, right? So it's not going to degrade, but it's also going to be stable physically. And so that means that if the molecule's in the crystalline form, that it stays in the same crystalline form. On the other side of the spectrum, if the molecule is in the amorphous state or the non-crystalline state, that it remains in that state. And it's that sort of physical stability that becomes and can become very important, both to the chemical stability as well as to the solubility of the molecule. And then, when you develop your product, got the right chemistry, and you're able to solubilize your molecule, and you've got the stability that's required, then you need to make sure that your product is manufacturable. And so that's, again, why it's important to be choosing chemistries and unit operations that are well known and well characterized, not only in the industrial pharmacy, but also in the company that you work for. And you can span the breadth of chemistries and technologies from spray drying and hot melt extrusion to lipid-based systems that are in soft gels, all the way to surfactants and nanomilling that are typically used for capsules and tablets.
So you've alluded to some technologies, and could you elaborate on the technologies that are used to enhance solubility and therefore bioavailability, and what factors go into selecting the right technology for a particular API?
For example, let's choose lipid-based systems and soft gel, and we can move on to some others like hot melt extrusion or then even particle size reduction and nanomilling. But the first and primary concern is to find a technology that you have the expertise to be able to utilize. The second thing is you want to also then be able to use a technology that has broad applicability. And the third thing, as I had said before, is you want to then make sure that your product is manufacturable, which means not only can you make your pilot scale batches and then your demonstration batches, but you're going to be able to scale up the product, validate it, and then reliably supply that product. Some of the very well tried and proven technologies, lipid-based systems, for example, that can go into any one of a number of encapsulation technologies, but in particular soft gel is certainly well proven. Over 50 NCEs are in a lipid-based system, and that number continues to grow, in part because you've got this incredible range of lipid-based systems or oils that can be used to solubilize a drug. And quite simply, like dissolves like. So if you have a poorly soluble drug, then it's going to want to be in something that's also poorly water soluble, and so that means it's very likely to be soluble in an oil. And you get some additional benefits to having the product already solubilized in a solution when you're working with a lipid-based system. That, again, you don't have to deal with tablet dissolution and disintegration as you do, with other solid oral dosage forms that you don't have to be concerned with for a lipid-based system. Hot melt extrusion is also another technology that's seeing more adoption these days, and I think primarily what's driving that is, the scientists who are formulating these products are becoming more and more comfortable with the chemistries of the excipients that are necessary to solubilize the drug and then also maintain stability. And then I think, classic is the, size reduction techniques, and whether you're going to use, media milling or you're going to use some sort of jet milling technology. That's also another good, well understood, straightforward way to really understand what can be a simple but elegant way to improve the bioavailability of a molecule. So kind of to summarize a little bit, Patricia, there's things you can do physically by controlling the crystalline state or lack of a crystalline state. You can reduce the particle size, and again, those are two techniques that have been used for many years and will continue to be used for many years. And then you've got lipid-based systems like the soft gel, again, been used for many, many years. And we're starting to see hot melt extrusion used a bit more as well. And we'll likely even see some more products that are being approved with spray drying as well. But I'd say almost all the time a formulator or medicinal chemist is going to work with what they know, and that's going to be the lipid-based systems and the hot melt extrusion and then the particle size reduction techniques.
Can you take us through an example of the decision points used to select the right solution for a poorly water-soluble compound?
Well, when we talk about decision points, I think what we're talking about is how is the product performing? Do you have a food effect? Do you have a challenge with reproducibility of the blood levels? And I think a good example of a product where those kind of challenges were overcome was cyclosporin, Sandimmune, and Neoral. So the short story there is, the product was originally approved as safe and efficacious for treating patients post-organ transplant surgery, and it's an immunosuppressant. So you could fairly easily imagine that you want to have a product that is convenient to give the patient and is very reproducible and consistent in its performance. So the product as it was approved was in a oil, was dissolved in an oil, and there was some variability in the blood levels, as you might expect, and there was also a food effect. The second generation product, basically eliminated both of those challenges by going with a more sophisticated lipid-based system where it's a self-emulsifying drug delivery system where the drug with the oils and surfactants that's in the lipid-based system, in this case a soft gel, when dosed, emulsifies and solubilizes the drug, increases the solubility to the point where it's practically the same as in the fed state. And so therefore you don't have the solubility difference that with the self-emulsifying drug delivery system that you do with a simple oil system. And so you get that benefit with using that system. Then also, another benefit is the reduction in the variability of the blood levels, really all driven by a much more reproducible and controllable way to solubilize the drug.
Okay, well, thank you very much, and thank you for joining us today. This has been Patricia Van Arnhem, Executive Editor at Pharmaceutical Technology and Pharmaceutical Technology Europe.